Good morning, everybody. My name is Victor Martins dos Santos. I'm a professor of systems and data biology at the Wageningen University in the Netherlands. I prepared this presentation with Dr. Rita Volkers, and we will focus on the build and test aspect of the DBTL cycle. We go now on, we have an another biotechnology, we want to produce a particular compound, X, and we have certain raw materials. So usually the question is, how do I get to my product X from raw material Y? So we need to look into the dependencies. There are thermodynamic dependencies, biological dependencies and constraints. So the question is, how do I get central question in this about technology is how do I get to my product X starting from a certain raw material Y? Usually we need to take into account a number of dependencies that I show here, thermodynamic, biological, economical, and operational. And given these dependencies, what are the DBTL procedures and standards that have to be developed and applied to have specifications that allow us process development? This is a question a normal question in bioprocess engineering. Now, to remind the DBTL cycle where people have, one has industrial specifications, one uses computational workflows, model-driven designs, and data analysis to design our process. That is what Dr. Nogales just showed. And from there, we want to build this, either protein engineering, rational strain engineering, automation, and test. And it goes in through the process until we get to our product. Now, we want our product X. So we have a list of specifications. We want, for example, to be water soluble, soluble to high concentrations, and not toxic to the host. This is a particular product, can be anything. Now, we also have a bacterial host. Often we have a host that we either decide to use because you know it, or because we know that this particular host will be very good. And usually we have also a list of wishes for the bacterial host, for the chassis, which is to grow, for example, at moderate temperatures, to grow fast, to be metabolically versatile, not having too complicated nutritional needs, being, for example, aerobic, easily genetic accessible, and if possible, well studied so that we understand what is going on. Now, we have our host and we have our product. The problem with the host that we have, and it has native pathways, is that we have no product X, because if we use glucose, say, or raw or glycerol, which are our starting materials, we see that the microbe uses these for their own purposes, so no X. So basically, what we do is build upon the design proposed by the design phase by using, for example, methods that Dr. Nogales just showed. We have a specific design. This is one picture that he showed. We have raw materials, glucose, and glycerol, and have the product that we want to get in the end. So what we have here, I just did this quickly because I want to show that we have two materials. These are native pathways that I just showed in the previous slide, and our designs tell us that if we want to have a product X, we do need to have knockouts here and the gene A and E, because if you want to direct it here, we cannot let it go that way. And if you need to have the product being exported, for example, to get out of the cell, one needs to implement a transporter system into the system. So if you go into the design, we see that we have to knock out these two gene systems, and we need to make knock-ins of these three systems, these two enzymatic systems and one transport system. So this is a little bit of design from design step, and this is something that the builders have to have in mind as well, to see does it make sense, because one needs always to interact with the designers, because there are many designs. One has to see what the designs are most appropriate and feasible. So we have the design tests, the design inputs, three knock-ins and two knockouts. Now we need some information transfer from the design process to the build process. So we need, of course, the name of the product, the properties, solubility, toxicity, you name the host, the various features of the host, the specific genes to knock in and knock out, or maybe more than just genes and knocking it out. It could be whole pathways. We need the pathway information, reason for selection of those genes, and so forth. Now, we want to start building. So we need to search for additional information and materials. For example, do we have the strain? Do we need to order it? Um, where to get those genes for knock-ins, for example? Do we have available, and are we able to use specific DNA manipulation techniques for the host? For example, what techniques need to be developed? What do we have in our own lab? What we need to learn and acquire. This is very important. We need to have, of course, the chemicals, the plasmids and constructs available, consumables, and so forth. And of course, the builders have to design as well. After selection of specific techniques for knock-in, knockouts, CRISPR-Cas, homologous recombination, and so forth, we need to select the plasmids to use, promoters, terminators. So we need to decide reducible, constitutive, how strong they are. We need to modify them, adapt them. We need to design the cloning process, decide what goes where in the plasmid, design primers, design DNA sequences. We may need to do codon optimization, a number of things that people do currently in the labs. But this is put into let's say, systematic workflow. This is what we want to do. In this particular case, where do we start? Well, usually it's good to start where the first bottleneck is, because if the first bottleneck cannot be overcome, then it does not make sense to do the other ones. Okay, so in this particular case, we want to eliminate genes A, D, and B, D, so doing the knockouts. We start by doing this, and once we do that, I'll come back to the second. We also have to say, suppose this will be successful, what would make sense to do subsequently? Now, if we start to have, if we want to produce X, we need to know whether the X, compound X, is toxic for the cell inside the cell. So if it's not toxic, you can proceed with the knock-ins. If it's toxic, we do need to provide a transporter for this. So this is still part of the, let's say, the design phase of the building process. Then to make the mutant strains, this is actually a two-step process after ligating and other parts of the plasmid to transform and clone in a host. For example, E. coli, check sequencing plasmids, multiply plasmids, and so forth, multiple plasmids, and so forth. We need to transform the plasmids into the host strain, and we need to check if the strain is correctly mutated, often through sequencing. Let's say we start with step one. We have knockout of the gene BD of this part of the pathway, of the native pathway. We use CRISPR-Cas, for example, or homologous recombination. We implement this. We look to, once we implement this, does it work? It means, do we still measure compound D? If you don't measure it, possibly the knockout step has worked out. If not, we have to troubleshoot. I come this into us in a second. Now, step two, again, we want to use knock out the genes A, E. We use again the techniques that we had available, CRISPR-Cas, homologous recombination, and other could be done together with step one, depending on the technology. And this can be done subsequently, and of course we need to then to check, well, is this compound there? If it's not there, then we are in a good condition. We think we can proceed to step three. If there is still compound available, we need to troubleshoot. Okay, and what it means, troubleshooting means we need to see, is that a technical problem? For example, the knockout process did not work and the genes are still there, or it could be that the genes have been deleted, but there are, for example, isoenzymes or other enzymatic systems that carry out the same reaction that we do need to know about it. So we need to go back to the design, analyze, for example, through all of the genome space and see, do we find are there any indications of genes that can carry out this function? And we need to do this iteratively until we get our process of doing the double knockout. Now we go to the third step, which is the knock-ins. We have to introduce those sets of genes A, C, and CX to make sure we have our compound X, and then we need to have a transport system to make sure that this compound X gets out of the cell at sufficient rates. So we need to use then techniques such as overexpression on a plasmid, integration in the genome, either random or with a specific location. We need to make a number of choices, whether all genes are an operon or each gene is its own promoter. This is all types of, let's say, little design steps that one has to have within the build process. Okay, now then again, once we implement this, we suppose, of course, that we are able to do that. And if not, we need to have the troubleshooting as indicated. We need to test again. Now, if we have our compound X out, we should be happy. We can proceed to optimize the process and enter, let's say, the optimization scaling gate phase at some point. Otherwise, we need to troubleshoot. And by troubleshooting means, again, we start to analyze the internal concentrations of C and X, and we see, well, if we need to see if we need to add C to synthesize X, or otherwise the gene AC is not working. We need to see whether gene DX is not working. This is, let's say, the analysis of the problem initially. Solution is, of course, to optimize gene expression, or, of course, depending on the conditions, the optimize gene expression, looking at different promoters, terminators, optimization, codon optimization, or we just need to find alternative genes. This is, of course, a lot easier said than done, as many of you know, but it's important to have this in a systematic manner. All right, so once the mutant host is working correctly, that is, we get out our compounds X out of the cell, we have brought in the knockouts and the 3 knock-ins, and then we have to test it. So we know that it's working. In a sense, we already tested that it's working, and then of course what we want to see is, now, can actually these be produced in sufficient amounts? So typically, people would start to culturing the modified microbe in a standard medium in aromite flasks, culturing in medium that has been itself proposed by the designers, because in many cases there is quite a deal of medium optimization to be done to make sure that this does not limit the growth of the host. And this now has to be done, let's say, with multiple times and correct controls to make sure that have sufficient optimization. So we would have, we need to proceed with testing. There are variables that one has to look into, say temperature, medium composition, the volumes, the carbon source concentration, combinations thereof, continuous cultures, very batch cultures, the shaking speed, oxygen concentration. These are many of those conditions are more operational conditions, but we need to look into them after we have our catalyst working properly. And of course, we want to measure, there are many things that have to be measured. We want to look into the product concentration. We look into the substrate to the carbon source concentration, the various intermediate metabolites, because we want not only a chassis that produces lots of X, but we wanted also to produce only X and not X plus another 10 other compounds, because otherwise the process would be useless. We use for that a number of analytical techniques, HPLC, LCMS, GC, and so forth. That depends on the specific set of products that we're looking into. Now, in many cases, we need to proceed to high throughput testing, for example, using mini bioreactors such as a biolector, microtiter plates, because in many cases, although we achieve certain productivity, this is by far not sufficient for an industrial process. In most cases, just a little bit, and this needs to be highly optimized to have the possibility of being considered for possible industrial application. So it means that normally one has to undergo the system into evolution and adaptation procedures, in many cases also using mathematical models to help us on that. We need, of course, to screen colonies. There's a lot of robotics involved, flow cytometry, cell sorting. This is not strictly necessary, but often if one needs really to optimize a process, this plays a role. And this by being able to do it high throughput testing, one can change quite a number of variables at the same time, because this is what it entails optimization. And that means temperature, medium composition, volume of the culture, carbon source combinations thereof. Then, of course, we have continuous very batch or fed batch cultures, shaking speed gain, oxygen concentration, and so forth. So these are steps that relate to the testing and optimization. And after high throughput testing, we go into a slightly larger scale to lab scale fermenters, often to up to 1 or 2 liters, where one can have, for example, 16 such fermenters being operated in parallel. That's already quite some effort to be able to do that, and one does it only when one is certain that the compounds are being produced at rates that are promising, sufficiently promising for application. And this will be then a step prior to downstream processing and scaling up, which will be the subject of the next presentation. So on testing, one of course has to be done in collaboration with designers. In a sense, we have the designers, and then we have the builders and testers. But this is of course not just these are not silos. There should be sufficient communication between the various steps. We need to see, well, first of all, the results of testing have to be reported continuously to the designers, so that designers can optimize the process and use computational models to refine predictions and help on generating better designs, so that the designers, builders, and testers can work fast and efficiently. Now, changes that could be proposed by designers, for example, in this iterative cycle could relate to the improvement of the concentration of the compound X out of the system. That means codon optimization, could be additional genes, could be knockout additional genes, could be a complete change of pathway, could be a change of cultural method circumstances. And this is in fact a much more common procedure of having to refine the whole process of design, build, and test quite substantially. That's why it's a cycle. So that there will be a limit, there will be a go, no-go point where one decides the process is sufficiently efficient and effective, and we want to proceed to a optimization and scaling up phase. So in the end, that's what one wants to have, is a result with high titers of compound X, and we want to have only compound X and not compound X and another 10, as I mentioned this before. So this is a summary of the build test step of the DBTL cycle, and we start by the design that was shown by Dr. Nogales, and we'll move now to the next presentation, where we'll deal with a whole case study and looking into the upscaling. It's important also that for this purpose, and also to make sure that all of the data generated, the many types of data generated, can be stored, can be managed, can be useful for further development. It's important to have in place solid laboratory management systems, so that all the information can be analyzed in a fair, can be analyzed and deposited and managed in a fair way. So this provides a little bit of a scheme on how to put all this information into a data storage framework. So I'd like now to thank you for your attention, and I would be open for questions. So do send me an email to this email here, and I'll give the word back to the moderator.